protein synthesis. Living cells devote their 80% of energy in protein synthesis. An overview of translation. Translation needs 4 to 5 main components. The first is tRNA, the second is ribosome, third is mRNA, and fourth one is enzyme amino acyl transferase. These are the four components or important components of translation. Now the ribosome, they translate mRNA. See, out of 3 RNAs, only mRNA is translated into protein. The mRNA is translated in 5' to 3' direction, producing N-terminal to C-terminal polypeptide chain. Okay, that is, the proteins are synthesized from the N-terminus to the C-terminus. Amino acids are linked one by forming peptide bond between two amino acids. C-terminal of first amino acid is linked to next amino acids N-terminal or amino terminal. N is amino terminal and C is carboxy terminal. So amino acids bound to tRNA are inserted in proper sequence due to specific binding of each amino acid to its tRNA and specific base pairing of tRNA to the mRNA codon. Okay, the tRNA has anticodon and mRNA has codons present on it. So these two are complementary with one another, and the binding facilitates dropping amino acids into the chain for synthesis of polypeptide. Components in translation are mRNA. In eukaryotes, mRNA is made in the nucleus and transported to cytoplasm, and in prokaryotes, transcription and translation occur concurrently, occur simultaneously. As mRNA synthesis starts, immediately protein synthesis starts in prokaryotes. In eukaryotes, mRNA is synthesized, it is modified, transported to the cytoplasm, and then translated. Remember this difference, important difference of prokaryotic and eukaryotic mRNA. tRNA serves as adapter molecules that mediate the transfer of information from nucleic acids to the proteins. tRNA serves as amino acid adapter. They bring amino acid for synthesis of peptide chain. Ribosomes, these are units, manufacturing units which synthesize proteins. These are located in cytoplasm and these are made up of rRNAs and proteins. rRNAs and proteins, they form ribosomes. Enzymes required for translation are the one which charge tRNA. Charge in the sense attaches amino acid to the tRNA, and the other enzyme is for peptide bond formation between amino acids. Then the proteins, these are soluble factors necessary for proper initiation, elongation and termination of translation. Okay, as we have discussed different factors needed in replication and in transcription, same way here are the factors involved in different stages of protein synthesis. Initiation factors are labeled as IF elements, IF initiation factors. Elongation factors are EF and termination factors are TF. The tRNA molecule, it is the smallest RNA of a length 70 to 95 nucleotides. Okay, this tRNA have clover leaf shaped structure or L shaped structure, where few of the amino acids are few of the nucleotides present in the tRNA, they are modified after its synthesis. This tRNA has different loops. One is known as anticodon loop, which binds to the mRNA. The second loop is known as T-Cy C loop, and third is known as DHU loop. These two loops contain modified bases. The tRNA has inverted repeats present in it, so they form intra-strand base pairing with one another and form structure like this. Okay, the tertiary structure like this. The amino acids are attached to this 3' end. See here, at the 3' end, tRNA is modified by adding CCA triplet, CCA bases at this 3' end of tRNA. All the tRNAs have this CCA basis, and to this adenine last base, amino acid is attached. Okay, or to this second base, cytosine, amino acids are attached. Once the amino acids are attached, the tRNAs are said to be charged tRNA. When it is without amino acid, this tRNA is known as uncharged tRNA. Okay, this is anticodon loop, which consists 3 nucleotide sequence or codon, one codon, which is complementary to the bases present on mRNA. This anticodon loop dictates which amino acid have to be attached to the specific tRNA. Say here, this GCA codon present on mRNA codes for alanine. So this GCA binds with the anticodon of tRNA. Now the anticodon of tRNA, these two bases are fully complementary. Third base pairing may not be complementary. Okay, so according to this anticodon loop, the tRNA amino acid is attached. Now this is going to bind to alanine codon, so this tRNA is charged with the alanine amino acid. Alanine is linked to the 3' side of the tRNA. Okay, charging requires ATP, and the enzyme aminoacyl tRNA synthase attaches amino acid covalently to the 3' end of mRNA. Okay, so the recognition of codon is important for tRNA. So are there tRNAs available for every codon? So there are 64 codon in total. Out of this 64, 3 codons are stop codon. So the 61 codon out of 64 codes for different amino acids. So actually there must be 61 tRNAs available for each codon, but this is not the case. There are about only 20 to 40 tRNAs available. So how these 20 to 40 tRNAs bind to 61 codons? This is the base for Wobble hypothesis. Wobble hypothesis states that the third position of anticodon is not fully complementary with the codon. Okay, so this base pairing may vary, and therefore the third position of anticodon is known as Wobble base. So one anticodon can recognize several codons. So the Wobble hypothesis, Wobble occurs in anticodon. The third base in the codon is able to base pair less specifically. It is not fully complementary because it is less specific and less constrained three-dimensionally. This allows tRNA anticodon to recognize 3 different codons. This does not obey complementary base pairing in certain cases. Here is the example shown. So this is normal pairing where CUC codon pairs with GAG anticodon of tRNA. And this is Wobble base pairing shown where CUU codon present on mRNA pairs with GAG. So this third anticodon's base may pair with any of the base. Okay, there are certain rules for it. Okay, so this is known as verbal pairing, and the third position is known as verbal position. So these are verbal rules, anticodons present on tRNAs. These bases may combine with these bases on mRNAs. G can pair with U or A, C pairs with G, A pairs with U can pair with A or G, and I can pair with U, C or A. So the enzymes, the amino acid tRNA synthetase catalyzes attachment of attachment of amino acid to the tRNA molecule. There is at least one amino acid tRNA synthetase for each amino acid. The attachment of amino acid activates or charges tRNA molecule, and hence the tRNA, when linked with the amino acid, is known as charged tRNA. Okay, charged tRNA. The attachment of amino acids is at its carboxy terminal. Okay, carboxy terminal. So the ester bond forms between amino acid and tRNA. Next enzyme is peptide transferase enzyme. This catalyzes transfer of peptide when growing by addition of single amino acid. This forms a peptide bond between two amino acids. This we'll discuss later. Okay, ribosomes. These are the sites for polypeptide synthesis, machineries for synthesis of proteins. One second. So the ribosomes are sites of polypeptide synthesis. They recognize features that the signal the start of translation. They ensure the accurate interpretation of genetic code by stabilizing interaction between tRNA and mRNA. They supply enzymatic activity for synthesis of polypeptide and for forming peptide bond, and they facilitate the linear reading of genetic code by sliding along the mRNA molecule. Ribosomes, they read codons present on mRNA, walk onto the mRNA, read each codon one by one. Now, ribosome has two subunits, one larger subunit and one smaller subunit. Prokaryotic ribosome is known as 70S ribosome. S stands for sedimentation rate of ribosome when they are centrifuged. Okay, prokaryotic ribosomes larger subunit is of size 50S and smaller is of 30S. There is no correlation between the total of this and the size of whole ribosome. Okay, and eukaryotic ribosome has size 60S larger subunit, 40S smaller subunit. Okay, the prokaryotic ribosomes larger and smaller subunit, they are made up of rRNAs and proteins. Okay, the number of proteins are different and rRNA types present in each subunit is different. In eukaryotes, rRNA synthesis and ribosome assembly takes place in nucleolus. Okay, because nucleolus is the site where rRNAs are synthesized. Before translation begins, two ribosomal subunits exist as separate entities. Soon after the translation starts, they come together and form whole ribosome. The role of ribosomes in translation, the small subunit initially binds to the mRNA. Now, how it binds? The sequence present on mRNA is complementary with the rRNA sequence present in smaller subunit of ribosome. Smaller subunit of ribosome contain rRNA. The sequence, few sequences of rRNAs, they are complementary with the mRNA sequence. So they are aligned. Ribosome smaller subunit is aligned onto the mRNA by forming hydrogen bonds. Larger subunit provides enzymatic activity like peptidyl transferase activity and catalyze formation of peptide bonds. The assembled ribosomes creates three pockets for tRNAs for binding of two tRNA molecules. These three pockets are known as E site, exit site, which binds to uncharged tRNA. The middle one is known as peptidyl or P site, and the third one is A site or amino acyl site. Okay. So this is 3D structure of ribosome. Smaller subunit, larger subunit, and mRNA. So see, this red colored one is mRNA molecule. This is smaller subunit of ribosome. This is larger subunit of ribosome, and these are the three pockets present in ribosome: E site, P site, and A site. Okay. Now see, first tRNA binds here in P site, which brings always methionine amino acid, which is charged with methionine. Next tRNA, so the ribosomes align in such a way that, or ribosomes join mRNA in such a way that P site, now see here you can see each site, E site, P site and A site accommodate one codon. Codon means three nucleotides. See, this E accommodates GGG, P accommodates UUU sequence, and A accommodates AGC. So the protein synthesis always starts from AUG codon. Now ribosomes binds to the mRNA in such a way that AUG is in P site. Therefore, initiator tRNA binds here in P site. Next tRNA, bringing the amino acid dictated by next codon, binds in A site. So peptide bond forms between amino acid number 1 and amino acid number 2, methionine, and second say glycine. Okay, peptide bond forms between two. Now ribosome moves one codon ahead. Okay, as it moves one codon ahead, this AUG initiator codon will be in E site, and this tRNA transfer its amino acid here onto this tRNA. So here this tRNA is known as peptidyl tRNA, okay, which carries peptide, dipeptide, because it has two amino acids linked together. This will be uncharged tRNA. It will move in E site as ribosome moves one codon ahead. Now this site will be empty because ribosome moved one codon ahead. Next tRNA will bind here in A site. Okay, this is exit site for uncharged tRNA, from where the tRNA exits, deacetylated or uncharged tRNA exits. So the translation is divided into three stages: initiation, elongation, and termination. Initiation sets the stage for polypeptide synthesis. Elongation causes the sequential addition of amino acids to the polypeptide chain as determined by the sequence of mRNA, and termination brings polypeptide synthesis to halt, stops synthesis of proteins. Now in initiation stage, translation always initiates by AUG codon. Wherever the AUG codon is present, that is the start codon, known as start codon, and translation starts from that point, that codon. So initiation codon is AUG. It is towards the 5' end of mRNA that is being translated. It is not the first 3 nucleotides present on mRNA. Remember, it is codon present towards the 5' end. It is not the first codon present on mRNA. To the left hand side or to the upstream side of the start codon, the sequence present which helps in binding of smaller subunit of ribosome to the mRNA. In prokaryotes, there is conserved region upstream to the start codon, AUG, known as Shine-Dalgarno sequence. This Shine-Dalgarno sequence is complementary to the 16S rRNA of smaller subunit. Remember, we are discussing prokaryotic translation. Okay. So the base pairing between the 16S rRNA of ribosome and Shine-Dalgarno sequence of mRNA helps in binding of ribosome onto the mRNA. Then mRNA scans for start codon and bind. See, this is 16S rRNA and this is Shine-Dalgarno sequence. Here is the start codon present on mRNA AUG. Okay. So this 16S rRNA and Shine-Dalgarno bind with one another, which helps in binding of ribosome smaller subunit to the mRNA. In prokaryotes, AUG is recognized by special tRNA known as initiator tRNA charged with methionine. This methionine is formylated. Formyl group is added to the methionine, and then this formyl methionine is attached to the tRNA, specific tRNA, based on its anticodon. Okay, this tRNA is known as F-MET tRNA. F-MET tRNA. Okay, once the smaller subunit binds, then it scans for start codon, and then initiator tRNA, that is the one charged with formyl methionine, binds. Then the larger subunit attaches to the smaller subunit to complete initiation process. In eukaryotes, small subunit binds to the methylated cap because eukaryotic mRNA is modified by 5' capping and 3' polyadenylation. So the eukaryotic ribosome binds to 5' cap. It then migrates to the initiation site, usually the first AUG codon. And in eukaryotes, the methionine need not to be modified by formylation process. So see, this is mRNA. To the Shine-Dalgarno sequence, this yellow color, smaller subunit of ribosome aligns. Okay, then these are the initiator factors IF1, IF2 and IF3. IF2 is associated with GTP. This IF3 brings initiator tRNA. IF1 binds to the smaller subunit, and IF2 GTP binds to the mRNA. I'll show you details with another example. Wait. IF1 and IF3 binds 30s subunit. IF2 GTP bind to smaller subunit, forming a complex at ribosome binding site. So first this IF3 binds to E site, IF1 and IF2 GTP binds to A site. When tRNA, initiator tRNA binds, this GTP, IF2 GTP is hydrolyzed to GTP, IF2 is released. Now the initiator tRNA binds in P site of ribosome. Okay, first tRNA binds in P site of ribosome. Then the larger subunit, and this is initiation complex forms. Then this initiation complex binds to mRNA. Then the IF3 is released, larger subunit of ribosome binds. Then IF2 GTP, once larger subunit binds, GTP is hydrolyzed to GDP, and IF2, IF1 both are released. See this GTP, when it is GTP, IF2 is active. When it is converted to GDP, when it is hydrolyzed to GDP, IF2 is inactive, therefore it leaves its place. Now this is initiation complex where peptide tRNA is present, initiator tRNA is present in P site. Now the next tRNA, next codon is in A site. Now in this A site, the next tRNA will bring second amino acid. Okay. See here, in P site, Fmet tRNA enters and amino acyl tRNA, AA tRNA, that is amino acyl tRNA, हेलो, हाँ बोला सर, हो ठीक है सर. So the next is elongation. These are elongation factors involved in prokaryotic translation elongation. EFTU, EFTS and EFG. All the three factors, they need GTP for their action. Three steps in elongation. The charged tRNA is delivered as the complex with EFTU GTP. That is EFTU GTP brings correct charged tRNA. Okay, accurate charged tRNA to bind into the A site. Then the peptide transferase makes peptide bond by joining adjacent amino acids without the input of more energy. And the translocase factor EFG GTP moves the ribosome one codon ahead along the mRNA, ejecting the uncharged tRNA and transfer the ribosome peptide from mRNA. Okay, EFTU brings charged tRNA, EFG GTP helps in translocation of ribosome one codon ahead. and EFTS is exchange factor for GDP to GTP. Now this EFTU-GTP brings charged tRNA and drops it in A site. Okay, A site is empty. In P site is peptidyl tRNA which carries the peptide of amino acid. Once the correctly charged tRNA is inserted into the A site, GTP is hydrolyzed to GDP and EFTU-GDP is released out. This EFTU-GDP is exchanged with GTP by EFTS molecule, and then again it is active to bring next charged tRNA to drop in E site. Okay. Now these two, this is peptidyl tRNA in P site which carries peptide, and this is amino acid tRNA in A site which carries only one amino acid. Now peptide bond is formed between the last amino acid of this peptide chain and the amino acid present on this amino acid tRNA. Peptide bond is formed by joining amino carboxy terminal and amino terminal of amino acids. These two tRNAs, they are rotated about one another and the peptide bond is formed. Once the peptide bond is formed, the peptide from the peptidyl tRNA is transferred onto the amino acyl tRNA. Amino acyl tRNA is this one. Okay. This is amino acyl tRNA, and this tRNA is now uncharged because this does not have any amino acid or peptide on it. Now EFG-GTP moves one ribosome moves one codon ahead with the help of EFG-GTP. Uncharged tRNA leaves the complex. This is now peptidyl tRNA will be in P site and A site is empty for joining next amino acyl tRNA. This is E site, this is P site, and this is A site. When the peptide is transferred from peptidyl tRNA to the amino acyl tRNA, this tRNA is now uncharged. So the ribosome moves one codon ahead so as this charged tRNA exits from the ribosome from this E site, and this peptidyl tRNA with the peptide will come in P site. Ribosome moves only one codon ahead. Okay, and now this A site is empty for joining incoming tRNA. This process continues till stop codons are encountered in A site here. UAG, UGA and UAA. Any of these three, when is encountered in A site, there is no tRNA available for stop codons. So here release factor binds RF1 and RF2. They bind and they hydrolyze the peptide from the tRNA. They detach both the subunits of ribosome and stops translation. Okay, this was about prokaryotic translation process. In eukaryote, the process is similar as prokaryotes, only the names of factors are different and initiation stage is little bit different. These are the differences in factors. These are elongation factors, these are initiation factor, and these are release factor in eukaryotes. Eukaryotic ribosome binds to 5' cap region of mRNA and moves along it for scanning AUG start codon. So this 5' cap small subunit binds, scans for start codon AUG. Once its AUG is in P site, larger subunit binds and translation process continues. You just have to remember the functions of each factor. Initiator tRNA with the EIF2 GTP binds and EIF3 4C and 40S binds to the 43S ribosomal complex. Pre-initiation complex is formed, and then it is recruited to mRNA. Then elongation, same as like EFTU, EFTS and EFG. Equivalent factors are found in eukaryotes, EEF1 alpha, EEF1 beta and EEF2. And termination, only one release factor, ERF. This small e stands for eukaryotic release factor. So this was about protein synthesis.